Assalamu alaikum, everyone. In the previous lecture, we started discussing the different types of cloning. So in this chapter, we have discussed so far DNA cloning, and in this section, we're gonna learn about cell cloning and organismal cloning, cloning a whole organism from one parent, or cloning a whole organism from one cell, from a single cell. Okay, and we discussed briefly the characteristics of stem cells, and cloning as a technique was first done in plants. So scientists always wanted to attempt cloning plants and animals from cells. So basically, the cloning we're talking about is to generate a whole organism from a single cell. So we have single cell, and this single cell will be used to generate a whole organism. This is what is meant by organismal cloning. Now, more than 50 years ago, scientists began to attempt cloning plants, and they were interested in knowing whether cells, once they differentiate, they will lose all genes. So basically, scientists, they didn't know if all cells within a whole organism will contain all the genes. So they thought that maybe cell differentiation might accompanied by the loss of certain genes. So we discussed differentiation in the previous section, and cells, when they differentiate, they will become specialized, and they have specific or specialized function and structure. And scientists, they were thinking that when a cell differentiates, it might lose some genes. So they wanted to know if this is true or not. This is why they wanted to test whether a single cell taken from a whole organism will have the capacity to generate a new whole organism. Okay, so a single cell taken from a whole organism, okay, if all the genes are not lost in this single cell, and this cell is differentiated, if this differentiated single cell can generate a whole organism, this means all the genes within this single cell are present and they were not lost during the process of differentiation. So the first attempt of cloning organisms from single cell was done in plants. This is an experiment done in the 1950s by a scientist named Stewart at Cornell University in the United States. In this experiment, Cornell and his colleagues, or his students actually, they worked with a carrot plant. So they took, so basically in this experiment, so let's call it so they wanted to clone carrot جزر, carrot cloning. So in their experiment, they took a current plant, okay, or a carrot root actually. So this is carrot root. Of course, we eat the carrot, we normally eat the carrot roots, of course. And they took some cells from this carrot root, some tissue. So they cut some tissue, okay? And they took some cells or small fragments, let's say, from this carrot plant. So small tissue fragments. Okay. And then these fragments were cultured inside a test tube. So a test tube filled with, so here we have, let's say, culture media. Culture means زراعة, medium. Okay, so we have these fragments are further fragmented to generate single cells. So these are carrot root single cells. Okay, and so basically in the culture media we have single cells from the root, and some of these single cells, of course, because these are root cells, they are differentiated. So they took some of these cells, and these cells started to grow. So they started to divide, okay? And then these cells were able to generate like an embryo, a plant embryo, with cells. So this is a large embryo, a plant embryo with cells, and this embryo was again cultured in a test tube inside, let's say, a medium, culture medium, and now we have some parts of the plant started to appear. We have, let's say, the green parts. Okay, so this is called the plantlet. Okay, and this plantlet is cultured inside a medium in a test tube. And then this plantlet is cultured in the soil, okay? So that we have a new carrot plant or root. Okay, so this is a whole new carrot plant. Whole means camel. Carrot plant. So this is the experiment done in the 1950s to show that differentiated carrot root plant cells contains all the necessary genes, or contains all the genes that enable them to generate a whole mature organism. Okay? So one major conclusion from this experiment is that all cells, whether they are differentiated, or the majority of cells, whether they are differentiated or not, okay, contain all the genes that are necessary to generate a whole organism. Now, the genes or the genetic material in this new whole organism is identical to the genetic material in the cell that was in the single cell that was used to generate that whole organism. Okay. So in plant, we start with differentiated cells, and now these cells must undergo a process called de-differentiation. So remember, these are differentiated cells, which means they are specialized in structure and function. They are root cells. خلايا جذر. Okay, root means جذر. Carrot, جزر. So a single differentiated cell must first undergo de-differentiation, and then it can generate all the remaining cells of the organism. So plant cells can be, let's say, mature plant cells or differentiated plant cells can de-differentiate and give rise to all specialized cells of the organism. And these types of cells that can generate a completely new organism, they are called totipotent. Okay. So we have a new term I want to introduce, which is potency. Potency is basically potency in cells, in cell biology, indicates the capacity of, or basically the ability, can be defined as the ability of cells to differentiate into specialized means differentiated cells or cell types. Okay, and there are different levels of potency. So totipotent, the highest level of potency is totipotency. Just like this cell from the carrot, this root cell from the carrot, which already differentiated, could be de-differentiated. And when it is de-differentiated, it will become totipotent. And once it's totipotent, it can basically generate, it can start dividing and then generates an embryo. Then it can give rise to all different cell types that are found in a new mature organism. So new, whole, mature. Mature means knowledge, kamil. All, sorry, knowledge. Whole means kamil. Okay. So plant cells, many mature plant cells, actually, they can become totipotent to give rise to a new whole organism. Plant cloning is used extensively in agriculture, zirah. So many plants, many types of plants, actually, cloning is the only way to reproduce them or produce them. Okay. This is in plants. Next, scientists, they wanted to attempt cloning animals from single cells. So it's more complicated in animals to do cloning. And because differentiated cells in animals or differentiated cells from animals, they cannot divide in culture like plant cells. So plant cells, they can start dividing in culture. Once they are de-differentiated and they become totipotent, they start dividing to produce embryo, then a plantlet, then the plantlet could be grown in soil. Okay, here we have soil to produce a whole plant. In animals, this does not happen. Okay. And scientists, they wanted basically to repeat this experiment to see if animal cells are also totipotent or not, or they can become totipotent or not. The approach used to answer this question was a little bit different in animals, because again, differentiated animal cells, they cannot divide in the culture. So what scientists did is that they performed something called nuclear transplantation. So nuclear transplantation, so again, scientists, they wanted to do cloning in animals to answer the following questions. Are differentiated cells or do differentiated cells have all the genes necessary to generate a whole organism? Or not? In other words, did any of the differentiated cell lose any genes during the differentiation process or not? And to answer this question, scientists performed something called nuclear transplantation. In this procedure, the nucleus from an unfertilized egg was removed and a nucleus from a differentiated cell was added. So this figure shows the procedure. So the first experiment of animal cloning was done in frogs. So scientists, they took a frog egg cell from females and they removed the nucleus from the frog egg cell by UV, for example, or by suction. And now we have enucleated egg cell. Enucleated, there's no nucleus. In one experiment, so in both experiments, so they've done this the same experiment. So this is called nuclear transplantation. Nuclear because they are transferring a nucleus. Okay. Trans means knuckle. Plantation, so they are taking a nucleus from one differentiated cell and planting it into an egg cell without a nucleus. Remember, this egg cell typically contains a haploid nucleus, one atom. And differentiated cells, they contain diploid nucleus, two atoms. So here we have diploid, diploid, here diploid. So the same experiment was done using two different differentiated cells. One is fully differentiated intestinal cell taken from a frog tadpole. Okay. The other experiment of nuclear transplantation was done using the nucleus of a less differentiated cell, a nucleus from a frog embryonic cell. So this cell is less differentiated than this cell. But both are differentiated cells, considered, let's say, differentiated cells. But this one is less differentiated. And both nuclei were added to enucleated egg cells. And this, now we have a zygote, or something resembles a zygote, or a diploid cell that can undergo development. And what happens is that the cell, when the nucleus was obtained from a fully differentiated intestinal cell from the tadpole, the embryo did not progress through later stages of the development, while the nucleus of less differentiated cell was able to support development of the embryo into the tadpole stage. Okay. So the take-home message from this experiment is that these cells, the differentiated cells, they retain their full genetic capability, which means they have these differentiated cells, whether from the tadpole or the embryo, they have all the genes that are necessary to generate a whole organism. Okay, but also this experiment indicates that the nucleus so the nucleus from a differentiated frog cell, whether from the embryo or the tadpole, can support the development of a tadpole. But its ability, the ability of these differentiated cells to generate a whole organism by cloning using this technique decreases as the cell becomes more differentiated. So the more differentiated the cell, the less likely that it will support or direct the development of a tadpole, for example. The less differentiated, the more likely the cell, the less differentiated the donor cell, the nucleus donor cell, the more likely the embryo will develop into a tadpole. A tadpole is basically one stage of during frog development. Okay. Now, why do you think less differentiated cells, the nucleus from differentiated cells, cell was able to allow the development of the embryo into tadpole compared to the fully differentiated cell, which was unable to direct the development into the tadpole stage? What do you think? What is the difference between the nucleus from this cell and the nucleus from this cell? What is the difference between them? Anyone? مش حدا؟ Okay. So again, the results of this experiment indicated that fully differentiated cells can direct the development of the frog embryo into tadpole, which means these fully differentiated cells, they have all the genes necessary for making a whole organism, and they do not lose any genes during the differentiation process. But at the same time, the results of this experiment indicated that the more differentiated the cell, the less likely its nuclei is going to allow the full development of the embryo into the tadpole stage. The less the differentiated cell from which the nuclei was taken during the nuclear transplantation, the more likely the resulting embryo will develop into the tadpole stage. Okay. So following the success in cloning in plant cells and the nuclear transplantation in animals, scientists began to play with the idea of cloning mammals, ثدييات. Okay. So scientists began to think about reproductive cloning of mammals, which is basically using the same nuclear transplantation procedure to generate a whole mammal organism from the nucleus of a single differentiated cell. Okay. And after many attempts, scientists were able to successfully generate a cloned sheep or a cloned lamb called Dolly. So Dolly was the first cloned mammal organism generated by using this nuclear transplantation procedure. And Dolly, this is Dolly. This is a picture of Dolly. And this is the parent donor. Okay. So we know typically in mammals, the generation of offspring requires the fusion of an egg with a sperm, because the sperm is a haploid cell, the egg is a haploid cell, and when a sperm fertilizes an egg, we have the zygote, and the zygote is basically a diploid cell, and this zygote will develop into a whole mature organism. So this is how, for example, mammals, including humans, because we are considered mammals, this is how mammals reproduce sexually. So this is sexual reproduction in mammals and many organisms, not just in mammals. Okay. تكاثر الجنسي في الثدي. Okay. So the procedure that was performed to clone Dolly was as follows. So they took mammary cells from a donor. So we have mammary cells from the donor, from one, let's say, parent. Okay. And they took these cells and they grow them in the lab inside a dish. Okay. So these are the cells. So here we have cultured. Cultured means مزروعة. Mammary cells. So mammary cells, they are taken from the sheep breast, خلايا ثديية, خلايا الثدي. Okay, and they grow them in cultured mammary cells cultured mammary cells. They grow them in, let's say, a culture media, but this culture media does not contain all the necessary, it's not a complete media. So it's so this is culture, or let me add it here, culture media, وسط غذائي. And these cultured mammary cells are actually semi-starved, مجوعة. So this culture media is not a complete culture media. It lacks some important nutrients. Why semi-starved? To allow these cells to, so semi-starvation or semi-starvation, تجويع, will promote these cells to de-differentiate. So semi-starvation causes de-differentiation, indirectly, of course. Okay. And then they took, of course, egg cells and from this cell they removed, from these cells they removed the nucleus. So here we have nucleus removal. Okay. So now we have enucleated egg. Enucleated egg without a nucleus. And then they took basically the enucleated egg and they fused it with the starved mammary cell, and the result is basically a nucleus, a zygote, basically with the nucleus from a mammary cell. Okay. So this is a nucleus from mammary cell. Okay. And of course, this eventually, this zygote, okay, and you have to understand this is a 2N nucleus, diploid nucleus. This zygote started dividing, producing or increasing the number of cells, and then this embryo was implanted in the uterus of a surrogate, we call them surrogate mother. And later on, Dolly the lamb was born. So implanted, so this is the embryo, الجنين, implanted, يتم زراعته في in the uterus of a surrogate mother في مبايض surrogate mother اللي هو أم بديلة أتوقع surrogate, I don't know what's the meaning of surrogate in Arabic, the exact word. So let me surrogate in Arabic means وقع أم بديلة أو تأجير الرحم أو الحمل البديل, something like this. So you can understand now that we have mammary cells coming from one sheep, egg cells from another sheep, and now a third sheep was used to carry the embryo and give birth to Dolly in 1997. Okay, Badil, yes, mother Badil. And after Dolly, scientists were, this procedure that I explained here is similar to the procedure done in frogs. It's called nuclear transplantation. This is the natural way of sexual reproduction in mammals, this is cloning a whole animal or a whole organism, or reproductive cloning of a whole organism from a single cell. Of course, following the successful cloning of Dolly, scientists have demonstrated that reproductive cloning could be done in other types of mammals like mice, cats, cows, horses, mules, pigs, and dogs. So, for example, CC was the first cat to be cloned. CC, short for carbon copy. But these, so here we can see CC, the first cloned cat. So this is CC, the first cloned cat. And this cat is called Rainbow. So the Rainbow basically is the nucleus donor. So Rainbow, a nucleus from differentiated cells from this cat called Rainbow was donated to produce Dolly. And as you can see, both cats are not identical. So look at the fur color. So here we have white, brown, and black. But CC has only white and let's say black fur. I don't know what's. So here we have orange, let's say, not brown, orange, black, and white. Here we have only, so genetically, they are both identical, but phenotypically, they are not, okay? Because there are some other environmental factors that are important in determining the final phenotypes of organisms. So cloned animals, they do not always look or behave exactly the same. In terms of the genetics, they are identical. So the same genes, the same alleles, they will have the same genes and the same alleles, but that does not ensure that they will look or behave يتصرف the same, okay? So even identical twins, so they are natural clones actually, they might look or behave slightly different from each other. Tala is asking, دكتور بس نعمل cloning مو شرط يكون نفس السبب. Yes, because you have to understand there are genetic factors and environmental factors. Play roles in determining the final phenotypic outcome or the final observable traits, okay? أو الصفات. Yes, so we will discuss the epigenetic effect in the next couple of slides. So let me stop here and we will resume the lecture at 12:30, okay? So to summarize, these two animals, the donor, the nucleus donor, and the cloned animals, In terms of the genetics, they are genetically identical, which means they have the same genotypes, genetically identical, متطابقين. And in terms of the phenotypes, no, they are not identical. They are different because you have to understand that a phenotype or a trait, the phenotype or traits, صفات, in general, they are determined by genetic factors and by environmental B influences. عوامل بيئي. Okay. So in the case of CC, so here, in the case of CC, we have also, because these are females, we have also random phenomena of X chromosome inactivation also plays a role. For example, so here we have genetic factors like we have genes or alleles, certain alleles. We have random phenomena like X chromosome inactivation and maybe genetic imprint. All right, so scientists notice that in most transplantation studies, only a small percentage of cloned embryos developed normally to birth. So you have to understand that Dolly scientists, they did this experiment around 226 times before they were able to successfully give birth to Dolly. So Dolly was the 227th attempt of this procedure. So scientists before having Dolly, they failed this procedure 226 times, if I remember correctly. So the percentage of success of nuclear transplantation was very low. This is one thing. The other thing is that these animals that were cloned, like Dolly, they were not normal. They look young, but inside, at the molecular level, their cells, they looked old. Okay? So you have to understand this, the nucleus that was donated to produce Dolly was a nucleus from an old mammary cells from an old sheep or old from old sheep, from old organism, let's say. Okay? So they did not live normally, no, ya Tala. Actually, Dolly exhibited defects, some diseases, and Dolly did not live a normal life. It looked young from the outside. From the inside, when scientists analyzed the cells of Dolly, they found that these cells, they looked like they are old cells. So Dolly died, I think, after 6 years, because Dolly suffered from a disease, and that disease cause the death, the premature death of dog. And when scientists studied why this procedure, the efficiency of this procedure was low, the efficiency فعالية. So this procedure suffered from low efficiency. Low efficiency فعالية قليلة, which means scientists had to repeat this procedure over and over to get basically to give to finally give birth to a cloned animal. And scientists that also the animals that are produced, they had high incidence of abnormalities. So not only this process is low in efficiency, but also the cloned animals, they had high incidence of abnormalities or defects. Defects يعني abnormalities أشياء غير طبيعية, defects خراب خلينا نحكي على مستوى التيشوز والسيلز والorgans. So now when scientists started researching why these animals, they had these defects and abnormalities, they found out that the cells from which the nuclei, the differentiated cells from which the nuclei was taken to for the nuclear transplantation procedure, they did not have basically a suitable epigenetic program. Okay. So we have something called the epigenetic program. So each differentiated cell will have a certain epigenetic program, which means we have in the differentiated cell, in the nucleus of differentiated cells, we have many epigenetic changes, such as acetylation or methylation, that will result in the differential gene expression. Okay? Because we know that histone acetylation or methylation will have an effect whether a gene is expressed or not. So the nuclei of differentiated cells, they will have certain epigenetic program, and they will basically express certain genes and they will not express other genes, or they will repress other genes. So in order to have a successful cloning, this epigenetic program before cloning must be reversed, meaning that cells, or the cells from which we take the nucleus, like here, their epigenetic program and differential expression program, okay, must be reset, okay, in order for the genes that are important for the development to be expressed during the appropriate developmental stages. This is one thing. Another thing scientists found out is that dolly cells, when they analyzed the chromosomes of of cells inside from Dolly, they had shorter telomeres. And we know that these shorter telomeres, do you know what are telomeres? So Dolly's cell, they have shorter telomeres. You know what are telomeres? Telomeres, anyone? Telomeres. Yes, telomeres are repeated sequences that are found. So we have chromosomes, for example. So telomeres are found at the ends of chromosomes. They are repeated sequences, and their job is to protect the chromosomes from erosion. You're talking about Yaragad telomerase. And these telomeres becomes shorter and shorter as the cell divides, and they become shorter and shorter as cells or as the organism ages. So the older organisms, they have shorter telomeres. So you could think of telomeres length as a measure of the age of the cells, okay, or the age of the organism. So the idea here is that although Dolly from outside looked young, like a young lamb, from inside at the molecular level, her cells were actually cells of an old sheep, okay? And the epigenetic, of course, this is because they had shorter telomeres, okay? Because they were taken from old organism or an old donor sheep. So these are the problems. So both the epigenetic programs or the epigenetic changes and the shorter telomeres, they had influenced basically the quality of life of Dolly, and they had basically an impact on the success, the low efficiency actually of the nuclear transplantation procedure we just discussed. The next topic, or the last topic, is about stem cells, خلايا الجذعية. And these cells, they are unspecialized, as I told you as in the previous lecture. This is the first property. And they can reproduce indefinitely, which means they can divide indefinitely, keep replenishing themselves. And under certain conditions, they can differentiate into one or more specialized cells. Now, stem cells, so as you can see here, this figure shows the three characteristics, major characteristics of stem cells. They are unspecialized or undifferentiated, or less differentiated. Okay. And they can divide to give rise to two daughter cells. And one of them could differentiate into different cell types. Okay. Of course, there are two major types of adult cells in terms of from where we basically get them, or in terms of their sources. So we have embryonic stem cells, or ESC, and we have also adult stem cells, a third kind of stem cells that could be generated in the lab, which is called induced pluripotent stem cell, or iPSC. In general, stem cells could be classified according to their potency into a number of cells. So again, one of the major characteristics of stem cells is that they can differentiate into one or more specialized cell types. So there is a system of classifying, so stem cell or stem cells classifications. تصنيف. So we have the class, we have cell type, and we have the mature cell lines generated from these different types of classes of stem cells. So the first type of stem cell, class of stem cells, are totipotent stem cells. So totipotent stem cells, again, we have a term called potency, which I explained earlier. Potency is the measure of the ability of a cell to give basically to differentiate into specialized cell types. The higher the potency, okay, the highest potency is for totipotent cells. An example of totipotent cells, we have the zygote, for example. Or we have some embryonic stem cells. And these totipotent cells, they can differentiate into any cell type. Into any cell type. So they can differentiate into any cell type. The next classification is for pluripotent cells, stem cells. They are they have less potency than totipotent. Some embryonic stem cells are totipotent, and induced pluripotent stem cells, also from their name, are pluripotent. They can differentiate into any of the 3 germ layers of the embryo. We have also multi-potent. They are less potent than pluripotent and totipotent. Okay. We have an example, the adult stem cells. For example, we have hematobiotic biotic stem cells. These are the cells that are responsible for making the cells of the blood. And these cells differentiate into limited محدود cell types. So for example, this type of stem cells can only differentiate into the cells of the blood, the red blood cells and the white blood cells. Next, we have oligopotent. Okay. Also, we have adult, some types of adult stem cells. These are some of the adult stem cells. They are found in organs, some of them. So we have, for example, the lymphoid, stem cell. Lymphoid stem cell. So the lymphoid stem cells, they give rise to certain types of white blood cells. So for example, they can only give rise to, they can differentiate into T cells or B cells and natural killer cells, but not all. So hematopoietic cells, they can differentiate into white blood cells and red blood cells and other types of blood cells. But lymphoid stem cells, they can only differentiate into white blood cells or certain types of white blood cells. Okay. So again, here we have also differentiate into more limited, let's say, okay, into more limited cell types. محدد يعني, limited cell types. And lastly, we have what we call unipotent. These are cells that can differentiate into a single cell type. Okay, we have some adult stem cells differentiate into a single one type of cell, single cell type. Okay. So as you go up like this, potency increases. So this is totipotent cells, they have the highest potency. As you go down, potency decreases. Okay. يعطيكم العافية يا جماعة.